Greetings, everyone. Thank you for coming to my talk. Uh, Firstly, I politely request that you imagine yourself in an enclosed space, like a house, isolating yourself, uh, quarantining yourself because there is a pandemic happening. You then find yourself feeling quite inquisitive. So you ponder the unanswered questions of the universe. You ask yourself, are we able to just stop the virus from replicating at the molecular level? Why, of all things, was toilet paper the first to be sold out? These are just some of the questions that keep me, Mark Nguyen, a third year undergraduate physics major, up at night. Having conducted cutting edge research under Professor Roya Zandi, a theoretical biophysicist here at UC Riverside, I will have the ability to share my knowledge about our ongoing research to examine the assembly and disassembly of the Calpia chloride model virus capsid. And hopefully, this will shine some light on one of those questions. So let us start off with some more questions. Like, what is the Calpia chloride model virus, or CCMV for short? And why are we interested in this virus in particular? So the CCMV virus is a plant virus. The cowpea part of the name comes from the fact that it infects the cowpea plant, or better known to many of us as the black-eyed pea, not to be confused with the band. The chlorotic part of the name describes the condition of plant leaves in which you may observe them turning a pale to dark yellow, as you can see here. Mottled simply means a pattern of irregular marks or spots. Putting this all together, we simply get that it is a plant virus that infects the black-eyed pea and leaves yellow spots. So you, at this point, you might be wondering, how does a plant virus affect me as a human? If it's only infectious to this specific plant, why should we even care about it? Shouldn't we be worrying about more deadly viruses, like maybe the one causing us to be under quarantine right now? Well, not to worry. We actually have researchers in our group studying this COVID-19 virus. But that doesn't mean that this virus that we're saying, the CCMV, doesn't uh, give us information about other viruses as well, and especially because there is an overwhelming amount of data for this particular virus. So let's get into a bit about what a virus is. I know everyone is familiar at this point, but let's go through the simple biology. But essentially, viruses are the simplest biological objects in essentially in biology. And they are composed of a genome, as you can see by those squiggles on the right, representing either DNA or RNA. And they have a capsid. Viruses have a capsid made of protein. Now, this capsid encompasses this genome right here. As I mentioned before, the capsid is made of a protein. So the CCMV capsid in particular has a specific feature that we are interested in. The interesting feature of the capsid holds great promise in enabling humanity to have a better understanding of the physical world, better understanding of virus replication, and better understanding of the way we can harness virus properties to advance the medical sciences. So you might be wondering, how does a physicist study a virus capsid? Shouldn't a biologist be doing that? Well, as a team of theoretical biophysicists, we are able to develop models that can accurately calculate and even predict the rate of assembly and disassembly of these capsids. Now, let me show you exactly what I mean when I refer to assembly and disassembly. Here in this short animation, you can see that the protein subunits are slowly forming or assembling around this, what looks to be like an orange sphere. Now, this orange sphere is the genome. Like as I mentioned before, it can either be DNA or RNA. Now, if you play this animation backwards, you have a rough representation of disassembly. However, for now, let us focus our attention on the way the protein subunits interact and work together to form this network around the genome. Using existing equations in physics, we are actually able to determine with strong certainty under what conditions, like protein concentration or binding energy, are required for this capsid to disassemble or assemble. These seemingly complex concepts can be broken down into, into relatively simple everyday analogies. Imagine again an enclosed room, except this time, fill the room up with balloons. Now imagine in this room, the four walls are have four high-powered fans. Now you turn on the fans, you can imagine that the air is pushing these balloons to collide with each other and collide with the wall. Now, this is precisely what we biophysicists observe and model, but on a much smaller scale. And instead of balloons, we're dealing with protein subunits. Now, to understand binding energy, I'm going to ask you to visualize one last thing. Remember when you were a kid and you would rub these balloons on your head and you noticed that it would stick to your hair? Well, now pretend that all these balloons in the room have been rubbed by your hair and they have this static electricity. Now you might start seeing the balloons clump together. This is exactly what we observe is happening to these protein subunits when we change the binding energy. Oh, you might say to yourself at this point, "Oh, wow, that's really interesting. But how does this assembly and disassembly physics promise all the advancements that you mentioned above?" Well, uh, let me tell you. So remember when I said the reason we are studying the CCMV capsid is because there is an interesting feature about it? Also, do you recall me saying that not too long ago that the orange sphere is representative of either DNA or RNA? Well, the interesting feature about the CCMV capsid, get this, is that there doesn't have to be DNA or RNA for this capsid to assemble or disassemble. And not only that, once it has disassembled, the process can be reversed and it can reassemble. The implications that this has is spectacular. Nanotechnology is the natural consequence of this in the form of what we call nano carriers. As you can see in this picture in the bottom left here, a nano carrier is essentially a very small drug deliverer that is usually injected into your body. If the act of assembling and disassembling of these capsids can be controlled at the molecular level, we are essentially able to open the capsid, insert a pharmaceutical drug, have the capsid reassemble, close up, and then have it disassemble if and only if it is at the site of the cancer cell, and then it releases the medication. Um, so the experimental Dutch researchers we are collaborating with actually noticed a special phenomenon about the CCMV when they recreated the CCMV capsid using what is called an elastin-like polymer, or ELP for short. As you can see here, these purple things inside these capsids right here. They observed the behavior at different pHs, at pH 5 and pH 7.5. They noticed that at the T3, so T3 and T1 simply refer to the symmetry. But for now, let's take T3, the bigger number is the larger capsid. The numbers essentially correspond to how big the capsid is. Let's say simplify that for now. And they noticed that when they put it in these different solutions, the T3 actually disassembled and reassembled into T equals 1, to a smaller size capsid. And this phenomenon has yet to have an explanation. Oh, sorry. Um, so uh, this is uh, this is um, this is where we theoretical physicists come in to tackle this research question, as you can see here. To understand and model this data given to us by our collaborators, we have a theory known as the classical nucleation theory, as you can see on the right here. So you can also see at this in this plot here that the T3s are assembling while the T1s are disassembling. And we want some kind of model or theory that can actually model that and encompass that. And thankfully, we have one by our very own Professor Roy Azani, developed this theory many years ago with the foundation, with the fundamentals of being able to model this exact data, this exact assembly and disassembly. So we know that by adjusting the protein concentration, the binding energy, and certain factors that change the rate of assembly and disassembly, we can essentially create a really accurate model for this plot you see here, and essentially revolutionize nanotechnology in the medical sciences. Well, thank you so much for listening to me today. If you have any questions, feel free to direct them. And uh, big shout out to my advisor, my mentor, who couldn't be here today, and a group member, Tony Montalvo, is actually in the group. She actually helped me with most of these plots as well. Thank you so much.
Wonderful. Thank you, Mark. I found that